Hello students, so this video is all about mitosis versus meiosis. So we're just going to start with a single cell, and I have used Lego pieces to represent the different chromosomes of the cell. And you'll notice that these two are about the same length, these two are about the same length, and these two are about the same length. Also, the fact that these three are red mean that they came from mom, and the fact that these three are blue meant that they came from dad. So what this means is this cell has two copies of each chromosome, and so we represent that as 2N. There's a red copy and a blue copy of the small, red and blue copy of the medium, red and blue copy of the large. And then we say 2N is equal to 6 because our grand total is 1, 2, 3, 4, 5, 6 chromosomes. So to start off with mitosis, the purpose of mitosis is to make an identical copy of this cell. So by the time we're done, we want to have two cells, both of which are 2N equals 6. Mitosis is used for growth, to add new cells to your body. It's used to repair a wound. If you're a single-celled eukaryotic organism, you might use this just to reproduce. And so we're going to go through the steps of mitosis, and the first thing that happens is the DNA has to be, well, actually the first thing is interphase. So in interphase, a couple things happen. First, there's a G1 phase where growth occurs. And since I've kind of represented this with marker, I can't make it grow, but the cell does grow. And then the second part of interphase is the S phase. S stands for DNA synthesis, and that means that the DNA copies itself. And we actually learned how this is done later in the year. You may remember DNA replication, where helicase comes in and unzips the DNA. DNA polymerase adds the complementary bases and builds the new strand of DNA. There's Okazaki fragments, DNA ligase, all sorts of things. That is a topic for another video, but all that's happening. So we're going to shortcut it. So let's just let these chromosomes replicate. You ready for this trick? Watch this. Wow, look at that. They have copied themselves. So note that in this, at this point, the blues have copied themselves, and they're connected by a little bit of DNA called the centromere. And when we're figuring out what the ploidy number is, 2N equals 6, you actually count the centromeres. And so in this example, 2N is still equal to 6. And this is kind of confusing, but there's 1, 2, 3, 4, 5, 6. Centromeres, and so we'd say there's 6 total chromosomes. So even though it's replicated and there's 2 identical copies, we would still say this is a single chromosome. Some other terminology, we would say that these identical copies are sister chromatids. And again, these two reds are identical. Now, the red and the blue are not identical because remember, this came from mom, this came from dad, and so we call these homologous chromosomes, meaning they carry the same type of information, but maybe slightly different versions. So for example, the eye color allele might be somewhere on this red chromosome, and maybe the red codes for brown eyes, but the same allele on the blue codes for blue eyes. Same type of information, but not identical. So we've got 6 chromosomes in homologous pairs, and the goal is to copy, make an identical copy of the cell. So the first thing that happens is in prophase, the DNA coils up and condenses. Now again, since we're using Legos for this, they're already condensed, so we can't really represent that. Remember, in interphase, the DNA is totally unwound and would be this kind of like a ball of yarn almost is what it would look like. So prophase, it would condense so you can see these distinct chromosomes. And then metaphase is next. Metaphase, think middle. And what happens is the chromosomes line up down the center of the cell. And you can tell I'm in trouble because there's not quite enough room to do it, but oh well. So metaphase middle, they all line up down the center of the cell, and in the real cell, they fit. And then of course this is totally random. So, you know, this could be here and this one could be down there. It's just all random. They all line up. And then anaphase is next. And what happens in anaphase, think anaphase apart, anaphase apart. The sister chromatids are pulled to opposite ends of the cell by the spindle. And so all of this is happening at the same time. They're pulled to opposite ends of the cell. And then in telophase, what starts to happen, check out my special effect here, the cytoplasm begins to pinch in two. And so basically these chromosomes are kind of on this side of the cell, these chromosomes are on this side of the cell, and then the cytoplasm begins to pinch in half. And then eventually cytokinesis occurs where the cytoplasm completely pinches in half. And now we have two different cells. Check it out. This cell is kind of what we started with. We had two small, two medium, two large, 2n equals 6. Our new cell, two small, two medium, two large. Lo and behold, once again, 2n is equal to 6. We have successfully made an identical cell to the original one. Okay, so now we are going to do a similar video showing meiosis. Now, mitosis, you remember, is to make a genetically identical cell. For meiosis, we have a very different goal. Meiosis is all about cutting the chromosome number in half and making the gametes, or the sperm and the egg. So to start off, we are going to start with the exact same cell we started in mitosis. So 2n is equal to 6, just like before. And then just like before, during interphase, the DNA is going to replicate. So let's do that again. Okay, voila, we've got ourselves our replicated chromosomes just like before. Now we go into prophase 1 of meiosis. Remember, there's two phases of meiosis. Meiosis 1, which is the separation of homologous chromosomes, and meiosis 2, which is separation of sister chromatids. So in meiosis 1, we're going to separate these homologous chromosomes from each other. And so the first difference from mitosis happens during prophase 1. What happens is these homologous chromosomes partner up. So they kind of join together like this. And then something called crossing over occurs, where little bits of DNA are exchanged between the homologous chromosomes. So what that means is this red and blue piece are going to exchange little bits of DNA all along their length. And when that happens, look at this, crossing over has happened. So you'll notice that little bits of red and blue have been exchanged all along the length of these chromosomes. Now I should have done the same thing on the medium and the large Legos, but I'll be honest, I didn't have enough Legos. Teacher salary problems. So crossing over is hugely important because this is a major source of genetic diversity. By crossing over, we're now sort of combining elements from the chromosome you got from your mom and the chromosome you got from your dad. And so you're making this unique combination of genetic material that has never before existed in the history of life on Earth, which is pretty cool. So a major source of genetic diversity is during this crossing over phase of prophase 1. Okay, so metaphase 1 is next. Just like mitosis, they're going to line up down the middle, but in the case of meiosis 1 here, they line up as pairs. Now, this is another source of genetic diversity because the way they line up is random. You'll notice I have the two blues together. That was just by accident. It could line up that way, or it could line up that way. It's totally random. And so ultimately, these are going to split and we're going to get, there's different combinations of genes that are going to end up in each cell because of the random nature of how these line up. So that is a second major source of genetic diversity during metaphase 1 here. Okay. Anaphase 1 is next, and in anaphase 1, again, they move apart, but this time it is the homologous chromosomes that separate, not the sister chromatids. So these go this way, and then telophase 1 and cytokinesis. Once again, I'll do my fancy trick where we do this, and then cytokinesis, they split. Now, here is an important little point. You'll notice at this point, we only have 1, 2, 3 centromeres. And same over here, 1, 2, 3 centromeres. So there's only 3 chromosomes in each. And you'll notice we only have the red medium, and we only have the blue long. And over here, we only have the red long and the blue long. In other words, we only have 1 copy of each chromosome. So instead of being 2n equals 6 at this point, we are n equals 3. These cells are now haploid. They have one copy of each chromosome. They have the long red, the medium blue, and of course, in reality, it's all a combination of both, but they've only got one copy, so we would say N is equal to 3. This is now a haploid cell. So in meiosis 2, basically what happens is we need to separate these sister chromatids. So prophase 2 happens, where the DNA will once again condense. I believe it unravels briefly in between, but not much happens in prophase 2. Metaphase 2, they line up down the middle, just like this. Anaphase 2, they're going to pull apart like that. And telophase 2, they're going to eventually split. I'll do it this way. They're eventually going to split into 2 different cells. And I'll do the same thing over here, where these are going to go to the other side. And so basically, by the time we're done with meiosis, we have now created 4 cells, each of which is haploid, and each of which has half the number of chromosomes as the original cell, and each of which has this unique combination of genetic material from both mom and dad. So we have created these gametes, the sperm and the egg, and we have made very many unique gametes because of the crossing over that happened and what's called independent assortment, which is how they lined up in metaphase.